Today we are going to learn about an area of microbiology without which the study of microbiology remains incomplete, and that is classification of bacteria. In my first lecture, I'll try to let you know the development and the early approaches to the classification of bacteria. If I take a gram of soil sample, there are millions of bacteria present in that soil. If the biomass of all the bacteria that is present on the surface of this earth is taken, we will find that it will be more than the biomass of all the plants and animals that are present on earth. This shows that we have got numerous amount of bacteria present on the surface of the earth, and they are in diverse forms. So, how these diverse forms are evolving in our environment? Our environment is constantly changing, and as with the changing environment, the microbes are evolving. That means the microbes are getting affected by the external factors of the environment, and then they try to compensate. When they try to compensate, some microbes are there which cannot compensate and they cannot maintain the homeostasis. As a result of this, they are lost from the environment and we lose the genetic makeup of that particular microbe. On the other hand, if the microbes are able to compensate with the changing environment, they retain the homeostasis, or they are able to maintain the homeostasis, and as a result we say that the microorganisms survive. This surviving microbe is not at all the same microbe which started its journey or which was exposed to the external environmental condition. Therefore, we have the evolution of some other form of bacteria or other form of microorganism. Now, when we want to study the classification process, we basically say that classification is an orderly arrangement of things. Say for example, if I say that I orderly want to arrange the number of students in a class, I can arrange them by means of their gender as girls and boys. I can arrange them by means of their merit, that is the percentage of marks they have scored, or either I can arrange them based on their height, I can arrange them based on their weight. So there are numerous methods by which the arrangement of things can take place. But whatever the arrangement of the things are, they have to follow certain rules and regulations. And these should be relevant, these rules should be meaningful, these should make a biological sense, and these rules should have an evolutionary context. As a result, we call these the consistent rules of all scientists. That means whenever a microbe is discovered, it should be classified according to the rules which should be uniformly maintained throughout the world. Now, in order to understand classification system, there are two important terms we should know first, and that is taxonomy and systematics. Taxonomy and systematics are very much interrelated terms, the basic difference being in systematics, we systematically arrange the things and they depict an evolutionary pattern. While in taxonomy, we do not find any evolutionary pattern in the arrangement. There are three important things that are considered in systematics. The foremost important thing is classification. If I take the vehicles on the road, I can classify them into different groups. Say for example as car, truck, vans or SUV. Now these are the classes of the groups into which the vehicles have been categorized. Now how they are categorized? They are categorized on the basis of some identifying features. Now, if I take the vehicles, the identifying features will be the engine size, the mileage they cover, or the number of passengers they can take, or the mode of transmission. Now, after identifying these characters, they have been grouped into different categories. But after identification, another important thing has to be considered, and that is called the nomenclature. Nomenclature basically means providing a formal name, and it should have two letters. The first one called genus and the second one called species or specific epithet. Now that means I am giving a formal name to the vehicles, as a result of which they are known by the same name all over the world, and this is called the formal nomenclature of anything. Now let us understand this classification, identification and nomenclature very detailed in the case of classification of bacteria. If I go back to the development of the classification system, we see that it started its journey in 1735 when Carolus Linnaeus divided the life forms into two as Vegetalia and Animalia. These were given the status of kingdom. Later on, Haeckel came and he said that we should have three kingdoms as Plantae, Protista and Animalia. That means inclusion of Protista took place, and this was because there were certain living organisms that lacked tissue differentiation. After this three kingdom concept, there was development of another concept where we say we could have only two categories, one as prokaryotes and the other one as eukaryotes. Prokaryotes which do not have a true nucleus and the eukaryotes which do have a true nucleus. Later on, Whittaker developed another classification system in which he classified the living forms into 5 different kingdoms, and we call it Whittaker's 5 kingdom concept, in which he placed a kingdom called Monera, in which all the prokaryotes, that is the living organisms without any true nucleus, were placed, and the rest of the 4 kingdoms were eukaryotes having a true nucleus. Later on in 1990, Carl Voss said that let us have another level which should be higher than the kingdom, and he named it as domain. And therefore, 3 domains in the life form were given, one called bacteria and archaea. Bacteria and archaea are the 2 domains which were placed in the kingdom of Monera, that was given by Whittaker, and the other 4 kingdoms were placed under the domain of eukarya. So basically, in our lecture, we will be concentrating on the classification of the domain which is called bacteria. If I go on to the classification of bacteria, that means I should have the different levels or the different categories and the groups into which the bacteria will be classified. Try to remember a very simple sentence and you remember the classification of bacteria. That is, King Philip came over for good spaghetti. That's a very good sentence. You remember it, and remember the first alphabet of each of the word gives you the different groups of the classification system. The K stands here for kingdom, P for phylum, C class, O for order, F for family, G for genus, and S is the species. So these are the different categories or different groups into which the bacteria can be classified. Now let us see how the classification system was started. What were the early approaches, how we could know that how to place the bacteria into different categories. So basically we say that 3 approaches were taken. One was morphological approach, the biochemical approach and the physiological approach. Morphological approach means when we study the bacteria by its morphology, by its phenotype, whether it has got the presence of cell wall, the flagella, the pili, capsule, slime layer. So all the morphological characters are taken into consideration and accordingly they are placed into different groups. This is called the morphological approach. In the second case, the biochemical approach, we study the biochemical characteristic feature of the bacteria. What enzymes they are producing, are there any specific proteins they have got, or how they are carrying out different chemical reactions within themselves. So these are the biochemical characterization of a bacteria, and when this is taken into consideration and accordingly they are classified, we call it a biochemical approach of classification. The third type, which is called the physiological approach of classification, in this we consider the different physiological processes that is taking place inside a bacterial cell. Basically, we refer to the metabolic processes, and accordingly when they are classified, we call it the physiological approaches. How this morphological approach and biochemical approach can be used for the identification, let us see, because we know identification holds a very important place in the classification system. I take a very common example. We call it an enterotube 2 test. Enterotube 2 is a tube. Here you can see this is an uninoculated tube where we have got 12 chambers. These 12 chambers have got 12 different media. That means 12 different media are meant for the growth of a single microbe. And here you can see there is a placement of a single needle. This needle is used for the inoculation of these 12 different media at single time. Now, if we inoculate this enterotube 2 and keep it at 37 degrees Celsius for 24 hours, then we could see there is a change of color. This change of color is now measured with the help of the numbers. Now, how do we do it? There are certain numbers given at the back of this enterotube 2. And if the change of color take place, we encircle that very portion of the number. After encircling those numbers, we get a value. This value is called the identification value or ID value. Here I am getting a number which is called 24160. After I get this number, I compare it with a reference table where we have different numbers given accordingly to different bacterial groups or to different bacteria. Now my number was 24160. As we can see from the reference table, this comes out to be bacteria which is named as Escherichia coli. So here we see with the help of a simple biochemical test, we can identify the bacteria. So this is what we call a biochemical approach. Now let us see another example where I just give you an example of a morphological approach in addition to a biochemical approach. Suppose I come across a bacteria and that bacteria is not known to me and therefore I label it as an unknown bacteria. If I stain this bacteria with a gram stain and I find it to be a gram negative bacteria, then I go on for the lactose fermentation test. If the bacteria is able to ferment lactose, then I get a positive result and then I further carry on the test which is called the indole production test. If the indole production test comes out to be positive, I categorize it into some other group, and if it comes out to be negative, it is being categorized into another group. Now let us see how this helps in the identification process. If the indole test comes out to be positive, I go on for another test, and that is called the citrate utilization test, where in the media I have got Simmons citrate. If the bacteria is able to utilize citrate as the carbon source, that is a positive test, then the bacteria is Citrobacter intermedius. But if the citrate is not utilized by the bacteria, that means it is showing a negative test, then it will be Escherichia coli. So if I have on one hand the biochemical test, that is enterotube 2 test, where I get the Escherichia coli, along with that if I have this morphological test in addition to the biochemical test, I can also come to the conclusion of Escherichia coli. Now if the indole production is not exhibited by the bacterial cells, then I can go in for the methyl red reaction. The methyl red reaction may show positive. If it is positive, then the bacterium will be Citrobacter freundii. But if it is negative, it will be Enterobacter aeruginus. So here we can see how the morphological approach along with the biochemical approach helps in the process of identification. Now after this identification process, the question came for the scientist is, how do we know that what test has to be done? How we can do the test in a synchronized manner in order to go to the specific level of that particular bacteria. So in 1923, David Bergey, he developed a manual and he called it Bergey's Manual of Determinative Bacteriology. As the name goes on, determinative. That means this manual is a sort of catalog where you are able to determine the genus, the species of the bacteria, to which particular group the bacteria belong to. This can be referred as the only reference book. 9 editions of this book have already been come out, but let us see what is nomenclature then. How do we name the bacteria after it has been identified or after it has been classified? Now, when we go in for the nomenclature, the first nomenclature was given by Linnaeus in his book Systema Naturae, and this nomenclature was known as binomial nomenclature. That means each formal name should have 2 letters. The first one is called the genus and the second one will be known as species or specific epithet. For example, if I take an example of a bacteria Escherichia coli, the rule of the nomenclature says that the genus name should always be capitalized. This E, the first alphabet of the genus, should always be in caps letter. The species name should never be categorized. The coli is the species name where we cannot use any capital letters. If both the names are used together, they have to be in italics or they should be underlined. Or the last rule says that if we write this formal name of the bacteria in the abbreviated form, we can abbreviate the genus with the capital letter, that is E, but we cannot abbreviate the species. So after all these informations about how to identify, how to classify, how to name the bacteria, this system of classification started. And the first system of classification of the bacteria that began its journey was phenotypic classification system. In phenotypic classification system, so basically we use the markers which were either phenotypic or the markers were biochemical markers. After this, we had the development of numerical taxonomy. Numerical taxonomy was a great help to the scientists because after the phenotypic characters and the biochemical characters were known, now a single bacterium showed hundreds of characters and it became very cumbersome for the scientists to carry out all those characters and compare it with another set of characters. So the computer was used at this time for the analysis of these characters. So basically what the computer did was they translated these characters into a value which the computer can read out. If the character was found to be present in the bacterium, the computer marked it as 1, and if it was not present in the bacterium, it was marked as 0. So by simply 1 and 0 method, the computer was able to designate the different characters to a bacterium. This numerical taxonomy very much helped us in the phylogeny of the bacteria. So this was the first approach towards the phylogeny, that is to see the evolutionary pattern of the bacteria. Because when computers were comparing the characters, they give the result in terms of percentage. Say for example, organism A and B are related 95%. That means they are nearby to the evolutionary tree. And when we find that bacteria C and bacteria A shows only 15% homology, that means they are not at all related. So numerical taxonomy was the first approach towards the evolutionary pattern of the arrangement or evolutionary pattern of the classification of bacteria. Therefore, Sneath and Sokal in 1983 developed the numerical taxonomy, which was great help to the systematics and to the taxonomy, and therefore helped the classification system to develop further. After all these informations in hand, David Bergey again collected all the results and said that we could have a manual where we could give the different groups of the bacteria into which they are classified. And therefore, he published another book, and that was completely different from the book of Manual of Determinative Bacteriology, but he named it as a Bergey's Manual of Systematic Bacteriology. So the term systematics was coming into development because now we are going to arrange the bacteria systematically according to the evolutionary pattern. This first edition of Systematic Bacteriology by David Bergey categorized the bacteria into 4 different divisions. The first division was called Gracilicutes. Gracilicutes includes all the gram-negative bacteria, those which were non-endospore formers and those which can be photosynthetic and non-photosynthetic. The morphology they exhibited can be rod, they can be cocci, or they can be curved forms. The second division that was given by Bergey was division called Firmicutes. Now, Firmicutes include all the gram-positive bacteria and they were non-photosynthetic. The third division was Tenericutes. In Tenericutes, he placed all those bacteria which show pleomorphic morphology. That means they do not have any fixed shape. They lack peptidoglycan. That means they do not have any cell wall. And the fourth group or the fourth division that he gave was Mendocicutes. In Mendocicutes, he took into account all the microorganisms that do have the cell wall, but the cell wall does not contain peptidoglycan. So, these were the 4 divisions given in the first volume of Bergey's Manual of Systematic Bacteriology. But the limitation of this manual was the bacteria were arranged according to their gram character. They did not show any morphological similarity because in one group we find rods and cocci, in some other group there may be presence of spirillum. So, the main criteria or the main character that was fixed over here was the gram character and no phylogenic evolution or evolutionary pattern was seen in the case of this systematic bacteriology. In our next lecture, we will continue with the second volume of Bergey's Manual of Systematic Bacteriology, in which we will see how the modern approaches to the classification of bacteria were taken into consideration.